So I'm going to talk to you about where gene therapy appears to be heading. And it's an interesting place right now because gene therapy is an area where there was a lot of investment, 2018, lots of money going in there, a cooling off of interest as the pandemic wore on, number of companies falling out, a lot of interest then perhaps in genome editing, in vivo genome editing. Some of that has cooled off. And we'll talk about where things may be going in the not too distant future. One of the key issues there we go. Just for those of you who aren't familiar with the major ways that gene therapy has been delivered, gene therapy basically two major ways of doing it right now. Taking cells from somebody and ex vivo modifying those cells with a gene therapy. That's basically how the chimeric antigen receptor T cells were made, and the first generations of genetically modified hematopoietic stem cells have been made. Or alternatively, giving a gene therapy directly, either locally, such as into the eye, or systemically. The major ways that these have been done, the gene therapy vectors for these two different applications, doing things outside of the body versus directly administering, have generally to date have been different sets of vectors. Modifications in vitro being done with lentiviral or retroviral vectors, in vivo gene therapy mainly with adeno-associated viral vectors. Adeno-associated virus is it's a virus that does not cause disease generally in people. But it can lose part of its genome. And so although it's a small, it can't carry very large pieces of DNA, and it can deal with any number of genetic diseases. It does have some challenges that I'll bring up in a little bit. The alternative way of getting things in systemically is to use other delivery systems such as synthetic capsids or lipid nanoparticles, and I'm going to focus a bit on that. We have 19 products and 20 different brand names of those products at the moment in the United States that are approved for gene therapies. The ones that are in blue-ish here are T-cell therapies that are modified ex vivo. The green ones are hematopoietic stem cells that are modified in vitro. And then the remaining purple ones are directly administered gene therapies, which cover now the directly administered gene therapies, things from a type of retinal disorder to hemophilia to Duchenne's muscular dystrophy. So I just want to talk a little bit about looking forward about where we see things going. So CAR T cell development was something that was developed over the past 15 years. A lot of it coming out of the University of Pennsylvania, but also out of Memorial Sloan Kettering. The current autologous chimeric antigen receptor T cell paradigm is you harvest white blood cells from the person, generally the person who has the malignancy that you want to treat. You then purify the T cells, activate those T cells, transduce them with the construct that recognizes the cell type you want to kill. In this case, it's usually B cells. And then formulate them and give them back to the person, usually after you've given them some more chemotherapy. This paradigm, it took Pharma quite a while to get this paradigm worked out. It's now done in the United States pretty much by two methods: centralized, which is the licensed products, or in a decentralized fashions by places like Memorial, Penn, making investigational products locally. And there's, we can, again, if you want to get into the, there's a lot of dynamic here about whether centralized versus essentially decentralized models are better here. I'm not sure whether that it's worthwhile talking a ton about that, because I suspect in 5 or 10 years, we will move past that to one of two things: either allogeneic CAR T cells, where these cells are engineered, so you take cells from a healthy donor, use genetic technology such as genome editing to essentially strip off the molecules that would normally cause rejection from an individual, and add in genes that you're looking to have the T cell recognize. The beauty of this is that with genetic technologies today, instead of just having one target, you can have multiple targets. You can essentially make using either CRISPR-Cas9 or other more standard technologies. You can make CAR T cells that essentially have Boolean logic. They need to recognize two things on a cell surface, but if they see a third, they won't become activated. And so this type of targeting may be the future. The reason why this is nice in the setting of being able to have a healthy donor where you can make 100 doses at a time is that it's more cost effective. Off the shelf is very nice. And potentially, if we start to think about expanding these products into uses outside of oncology, it would be a lot simpler, for instance, in autoimmune disease to have something off the shelf. It also may be that the perhaps decrement that you get when you have these allo CAR T cells in terms of persistence may be actually beneficial in the setting of autoimmune disease. The other place that I missed putting in my slide here because I was already anticipating I was going to run short on time, but I'll just say it. I didn't mention, but probably the next step for these that may be to get rid of the manufacturing facility altogether for making the cells and just give a CRISPR construct in a lipid nanoparticle or on a scaffold that directly targets the T cells and have essentially in vivo CAR T cell development. It turns out you don't need to get a ton of cells transduced with the gene of interest, but we'll see where that goes. The interest in gene therapy for rare diseases is that there are now estimated 10,000 plus rare diseases that need to be addressed in some way. Some of them are more serious than others. An example of a more common rare disease, sickle cell disease, beta thalassemia major. Recently, we've had gene therapies that have been approved. This is a genetically modified hematopoietic stem cell using CRISPR technology, using a non-viral method to get the CRISPR in, but has really shown that it can potentially transform these diseases. There's also a conventional gene therapy that does the same using AAV, but it's really remarkable that CRISPR technology, which the medical use of which was described in 2012 and led to some Nobel Prizes, is now actually part of an approved production process. CRISPR, first generation CRISPR was a nice molecule. It allowed us to do genome editing in a way that we couldn't before with previous genome editing technologies such as meganucleases. But the amazing thing is at Stanford and at MIT and other places in Boston, they have moved this so much ahead of where it initially was to having base editors, so you can fix a given base abnormality, any one of the 4 different ones you can imagine on DNA, or you can now read-write segments into the DNA or put cloning sites essentially into someone's DNA, so you can come along and put in a long segment of DNA. Those are prime editors, and the beauty of all of these is that you can actually deliver them in a different way than we do with most of our gene therapy. Because the biggest problem with adeno-associated viral vector gene therapy is that there are real challenges in making it at scale. It's a biologic process. You have to have a producer cell line. It's often a mammalian cell line. You've got to get all the things right, and then you have to get the virus particles made at where you like the virus particles to be chock full of the DNA vector that should be in them. And unfortunately, all too often, the production process leads to suboptimally filled capsids. So it's really a manufacturing challenge. Some people have gotten it. Some companies have gotten it spot on right. Others have not, and others who claim to have gotten it spot on right are lying. Sorry, I didn't have to keep people awake here, but they are. Because remember that FDA there is something, I feel like I'm a clinician at heart. FDA is a little bit like your doctor. They know what it looks like under the clothes. And so we know what companies are or are not doing in terms of their manufacturing. So we know sometimes we can't say anything. We know what they're saying publicly, and we know what's actually happening. But manufacturing challenges are a real problem. I'm also going to mention something about what we're trying to do at FDA to try to fix some of the clinical development timelines and different global regulatory requirements, because these things actually add a lot of time to development, which adds cost. Cost is problematic in two ways. It takes away money that could be going elsewhere, and it also, in some cases, causes companies just to leave the area, which is what has happened. And ultimately, it's all bad for patients. So we're trying to work on helping advance manufacturing for gene therapies, talking about how we can apply a platform approach, which may transform how we think about biologic products. And I'll tell you more about that. And then talk a little bit about some of the other more mundane things we're doing, like trying to maximally use accelerated approval, help getting to some global regulatory convergence, and helping sponsors interact with the agency more efficiently. It turns out one of the funny things about manufacturing AAV that may turn out to be helpful is that a lot of the process could be automated. It's just that no one has spent the time and effort, because the model that is not a great business model, although now we're trying to, and we're working with various grantees to try to think about a device type model for making the product, which is kind of a device with disposables, consumables to make small batch gene therapy. We'll see where this goes. I'm not going to hold my breath for AAV, because I suspect that over the next decade, we'll see AAV supplanted by CRISPR constructs. But AAV and other things, like even the mRNA vaccines, are platform technologies. They have some things alike, be they the vector backbone, the lipid envelope that carries them in, that are the same from product to product, and then you change something that gives it its unique specificity. So for gene therapy, it's the gene of interest. And it turns out that right now our paradigm is we make sponsors go back and do all the toxicology work. They have to provide all the manufacturing information, even though it might be the same for product 1, product 2, and product 3, and all the control information. So a couple of years back, working in consultation with Congress, we helped provide technical assistance for the Platform Technologies Act, which allows us now to designate something a platform for an approved product, and then sponsors can come in with subsequent products using that same platform where they modify it and just focus on the modifications and just essentially reference the platform product. We're actually also even doing this for products that are only under investigational new drug applications as well. But the reason why this is so important is that if we look 10 years down the line, maybe it's a little more, maybe it's a little less, I think we're going to see a transformation where CRISPR is going to be something that's going to be the workhorse of gene therapy. And if you think about CRISPR technologies, that prime editor to fix hemophilia A or hemophilia B, let's take hemophilia B, a rare bleeding disorder, which you can target the liver to fix the genetic mutation. That construct, and one that will fix a rare genetic disease that causes, it's a metabolic condition, hereditary hypercholesterolemia, by knocking out PCSK9, those two constructs are going to be 99.5% identical, with the exception of a stretch of nucleotides. And so suddenly our previous concept of what different products are starts to look a little different, because is it really, there's not a lot of difference between those two products targeting the liver, obviously for vastly different diseases. So do you regulate it by disease, or do you regulate it by organ or the type of mutation? So right now, I can't tell you we've settled on the way we're going to do it, but it's certainly not going to be what we're doing now because it's not efficient to regulate 10,000 products that way. It's probably going to be more reasonable to regulate this almost like a general tool in the device world, which is that it's not going to be regulated as a device, though. Don't. I'm not saying that. It'll be a biologic product, but you might have a genome editor that we approve for a variety of mutations that affect the liver in gene coding sequences or something like this. It's the reason why I tell you this is because I think that's probably where we'll end up seeing things move. It will require some real advances in technology because making these will require essentially small scale, high throughput machines, but we know how to make those. It's just, especially in this area where for these products, unlike adeno-associated viral vectors, this is chemistry. You make these things, you can deliver these as mRNA molecules. mRNA molecules you can make using DNA templates made in E. coli. You purify the DNA and you can just go to town and then you can make lipid nanoparticles. You can actually do it. That mock-up in the lower right is a system which is actually doing just this. It's taking RNA, encapsulating it in lipid nanoparticles, and you can do it in a reasonably small and efficient manner. Just turning to finish up here, I'm trying to keep us a little bit towards on time. We're also trying to do things that will help the ecosystem move faster. Now, accelerated approval is probably one of the most controversial things I could talk about. I have stones thrown at me routinely for use of accelerated approval. But it is a very useful tool to have in our regulatory toolbox because for many rare diseases and even some more common diseases, it's very time consuming to get to a traditional endpoint of how someone feels, functions, or survives. It's much easier to look at a biomarker change, such as does somebody express hemoglobin? Do they have blood cells or do they not have blood cells? That is, for instance, reasonably likely to predict future clinical outcomes. These use of biomarkers is something that is really facilitated in part by genetic technologies because generally, if you are giving a gene therapy, you know the gene product that you need to replace. You know something about the pathway, so you can measure something upstream or downstream in that pathway. And so it allows us to look at biomarkers that are reasonably likely to predict clinical outcomes. And honestly, you have kids that have a number of neurologic diseases that basically progress over the course of anywhere from 1 to 10 years to have a child who's developing normally to a child who needs total care. That's at age 10. And if you can intervene sooner by implementing use of accelerated approval, it may make a very big difference. And, you know, as long as it works, as long as we get it right, that 90% of the time, roughly, we get it right, it's okay. I mean, maybe sometimes a product that has a biomarker that looks good won't get confirmed out in clinical trials. That may be because the clinical trial wasn't done well or other things, or it may be because of the product, we had something wrong in the science. But we're hoping to do this. What's nice about this too is we can look at animal models, or sometimes there are human experiments. People who have 10% or 1% of a given protein or yet are normal, we then know that if we can replace that protein using a gene therapy to that same level, it's reasonably likely to predict that they're going to have clinical benefit. And the final two things I'll tell you about come from having had a front seat at some of the COVID-19 pandemic events. So Operation Warp Speed was, I think, despite some people's not believing that it occurred, it did occur. We actually did have Operation Warp Speed. There was something called SARS coronavirus 2. We still have SARS coronavirus 2. Operation Warp Speed managed to make a vaccine for this in 11 months, which was really record time through a lot of efficiencies. And some of those were regulatory efficiencies that we came up with. And one of the things that became apparent during this time was that we and other high-income country regulators were all doing the same thing. We're doing it independently. And in a pandemic sense, there's no reason to be doing that. We could all spend more time doing value-added stuff if we pool forces. So we're currently thinking, oh, maybe we could do this in a non-pandemic time and try to resolve some of the regulatory differences that we have and be able to streamline submission processes. This is particularly relevant for these small market products, right? If you have a market that there's only 50 people in the United States for that are potential candidates this year for a gene therapy, and you do an NPV calculation, the NPV calculation is not look good, okay, over the next few years, right? On the other hand, if you add the people in the European Union, and you have to probably adjust for what you're going to get reimbursed, but then add Asia, you suddenly start to have numbers, hundreds of patients that actually make an NPV calculation look more attractive. So if we can help make it so that a single submission can make it through multiple regulatory authorities without needing rework and without needing new experiments, that could potentially help us in this rare disease space. It actually could potentially help us in all disease spaces. We know it can work in the oncology setting because Rick Pastor in the Oncology Center for Excellence has managed to do this with about 8 or 9 countries for oncology products, but we're trying to do it now. We have a pilot ongoing with the European Medicines Agency because we've never done it with that system, which is a little bit different. For those of you not familiar with the European review process, they do use country rapporteurs rather than kind of the central review process that we use. So we have to make adjustments, but we're working through it. And finally, we're trying to help probably apply what I think was one of the things that, at least at FDA, we really contributed to speeding up vaccine and other therapeutic development with during the pandemic was we realized that, you know, normally with meetings in FDA about how you progress, we have these very formal type ABC and now D meetings where the scheduling happens, you send in a request, it gets categorized, and you get a meeting in 30 to 75 days. Then you get a reply from FDA, which is uninterpretable. You have to send back questions, and you go back and forth through this. I'm sorry, I was in industry, so I was sitting there on the other side. We agree with you. Okay. So what we did was we said, okay, let's change this. Let's change it up, and we'll give you a project manager's phone number and email. And when you have a question, just send the question in. If you have multiple questions, send multiple questions in, and we'll farm them out to the CMC reviewers, chemistry manufacturing control reviewers, clinical reviewers, et cetera. And by doing that, we managed to resolve so many questions more rapidly, and we saved a lot of just manufacturing wasted time. We were able to get things faster because we were able to say, people were actually willing to ask questions like, do you really need 3 conformance lots, or maybe because we're in the middle of a pandemic, could you accept 2 and we'll give you the 3rd during production? And so we did stuff like that, and it made a huge difference in terms of reducing time. So now we're in a place that we have to say, well, what was the cost of doing that? Well, the cost was it took extra people to do it. But it wasn't an incredibly large number of extra people. So what we're doing right now is piloting to say in the rare disease space to see, could we do the same thing for a couple of products in the rare disease space while having an external consultant measure the various times to milestones and getting feedback directly from the companies. And this pilot program picked these are all pretty bad genetic diseases that affect children. MGLY1, very rare, Rett's, Canavans, methylmalonic acidemia. They tend to cause essentially mental deterioration, seizures, and ultimately death. So we have these in the pilot. The pilot's going along. At least initial feedback is people like it a lot, but we'll see. We'll have to be able to show that this makes the difference. Now, the funny thing is, we actually did our own equivalent. It's not an NPV calculation, but the equivalent of if we actually put this in place for all rare disease products, what would it cost in terms of dollars? The amount is actually remarkably trivial in terms of the additional reviewers. It's somewhere between $30 and $50 million, which all it takes is like a couple products getting to market a quarter faster, and it pays for that. So we'll have to get the data so that during our next user fee negotiations, we may be able to see this expand. But I hope it works because ultimately our goal is to try to help this ecosystem move more rapidly forward. And I think this is going to be a really, at least as a regulator, I think it's an exciting time to be doing this because we're going to have to shift the paradigm. I think with genetic medicines, with these platforms, if we just continue to treat them like, oh yeah, it's another small molecule drug, it is another conjurer of it, blah, 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 it's going to be, we're going to go through. Has anyone ever seen the Mel Brooks movie, Blazing Saddles? You've seen the toll booth in the middle of the desert and they got to go back and get the nickels. It'll slow development down like that. So that's that kind of like the clip I show my team when I want to say, no, that's not how we want to do it. We want the express lane here. So that's what we're trying to get to. So thanks very much.